Hello students. Under the chapter reproduction in lower and higher plants. So today you have to study about structure of anther. So you have already studied in the 11th standard about the typical structure of flower. So before the study of this anther you have to know about the plural word of typical structure of flower. You know that the angiosperm, which are also called the flowering plants. So what about the flower? Flower is a condensed and modified shoot designed for the sexual reproduction. What is a flower? Flower is a modified and condensed shoot designed for the sexual reproduction. Now the flower is the more conspicuous part of the plant and which is also known as a site of reproduction. Site of reproduction now in this flower. To this structure of the flower consisting the floral whorls. You already studied in the 11th standard about this floral whorl of the flower. Calyx, corolla, androecium and gynoecium, these are the floral whorl. About the calyx and corolla. Calyx and corolla, which are known as a non-essential floral whorl because they do not directly involved in the formation of the sexual reproduction, but the androecium and gynoecium, which are known as a reproductive whorl, which are also known as a essential whorl because they are directly involved in the reproduction. So first of all, you know that about that this androecium. Androecium is the male reproductive whorl of flower, and now here the individual member of the androecium, it is known as a stamen. And now here in this stamen consisting the stamen that is made up of the three parts. Now first it is known as an anther, second is a filament and third about connective. Now in this diagram you can see here this anther lobe. This is a dithecous anther, that's why in the dithecous anther the four pollen sacs which are present there, that is known as a tetrasporangia. So now here in this tetrasporangiate type of the anther, four pollen sacs which are present here. Four pollen sacs means the four microsporangia which are present here. Now in this topic, under this androecium, you have to study the microsporogenesis also. Microsporogenesis means the formation of microspore or the pollen grains from the microspore mother cell, that is known as a microsporogenesis. And after this microsporogenesis, now this formation of this anther and all the structure of the anther you have to study about. So now here first about this. In the transverse section of the anther, this is the structure of the transverse section of mature anther. But in the immature stage of the anther, now before the formation of these pollen sacs, now the parenchymatous tissues which are present there and which are surrounded by the epidermis. Parenchymatous tissue, the some cells of the hypodermal cells of this parenchymatous tissue now differentiated into the sporangial cells. And this sporangial cell which also the divides into sporogenous cell and the epidermal cell, or which is also known as a parietal cell. This parietal cell form the wall of the anther and sporogenous cell which also they act as a microspore mother cell or sporogenous cell divides by mitotically and then form the microspore mother cell, and this microspore mother cell divides meiotically to produce the many microspores. So this is about the immature stage मध्ये, म्हणजे हे जे स्ट्रक्चर तुम्हाला आता दिसतंय तर हे न होता जेव्हा इमॅच्युअर असतो किंवा हे पोलन सेक्स फॉर्मेशन होण्याच्या आधीचे जे टिश्यू प्रेझेंट असतात ॲट द प्रिमिटिव्ह स्टेज ऑफ दिस विच आर द पॅरेन्कायमॅटस टिश्यू, त्यांच्यामध्ये डिफरन्शिएशन होतं, डिव्हिजन ऑफ लेबर होतं. डिफरन्शिएटेड टिश्यू झाल्यानंतर त्याच्यातले जे हायपोडर्मल सेल आहे त्या आर्चिस्पोरियल सेल प्रोड्यूस करतात आणि आर्चिस्पोरियल सेल डिव्हाइड्स झाल्या की मग त्याच्यापासून स्पोरोजिनस सेल प्रोड्यूस होतो आणि दुसरी होते परायटल सेल्स. स्पोरोजिनस सेल्स हे फ्रुटिंग सेल्स प्रोड्यूस करतात, म्हणजेच मायक्रोस्पोर मदर सेल प्रोड्यूस होतं आणि जी परायटल सेल आहे विच डिव्हाइड्स टू फॉर्म द एपिडर्मिस. or the wall of this anther. Now here in this structure, this is a mature structure and you can see the detailed structure of this. Now here this first of all you have to study about the wall of the anther. Wall of the anther, now outermost layer of this anther which is made up of the epidermis. So this epidermis which is made up of one layer of cell. And the cells are tabularly arranged, they are flattened and they are tabularly arranged also. Now this epidermis, it is a protective in nature. It protects the inner parts of this anther. Then below this or inner to this epidermis, then another layer which is present here, that is known as a endothecium. Now this endothecium which is also made up of one layer of cell, and this endothecium, the cells are flattened and these cells produces the fibrous thickening of callus. And these are cells which are hygroscopic nature also. That's why they help in a dehiscence of anther. This is the importance of this endothecium which help in a dehiscence of anther. Then inner to this endothecium, then 2 or 3 layer of cells which are known as a middle layer. Now this middle layer which are made up of 2 or 3 layers. This middle layer also help during the maturity, after the maturity of the anther. Now these 2 pollen sacs merge to one another, merge to each other and form a one chamber due to this middle layer. And at the maturity of this anther, now this middle layer which is disintegrated. So inner to this middle layer, now this is known as a tapetum. Tapetum which is also again made up of one layer of cell and which is also known as nutritive layer, because all the cells contains the cytoplasm, the dense cytoplasm, then vacuole, food vacuoles also, and that's why they provide the nutrition to the developing microspore mother cell up to the formation of the microspore. Then one important point that's very important related to this tapetum produces the sporopollenin. And this sporopollenin which form the wall of the pollen grain, that is known as a exile. It is the outer wall of the pollen grain which is made up of a substance that is known as a sporopollenin, and which that's very important. So now that is this tapetum also helps to the formation of the sporopollenin. It means that the exile of the pollen grain that is made up of the sporopollenin. So this is about this tapetum. Then inner of which, आता इथे जे प्रत्येक pollen grains मध्ये तुम्हाला दिसलेले आहे, so which are all the pollen grains आणि which pollen grains are arranged in a tetrad. But before that, before the formation of microspore or the pollen grains, microspore mother cell which are present here, and they are diploid cells or the which are all the sporogenous tissue, and each sporogenous tissue which also the act as a microspore mother cell and which divides by meiotically to produce a number of microspore or the pollen grains. म्हणजे एका जेव्हा एक मायक्रोस्पोर मदर सेल डिवाइड होणार आहे तेव्हा 4 मायक्रोस्पोर प्रोड्यूस होणार आहे किंवा 4 पोलन ग्रेन्स प्रोड्यूस होणार आहे, and they are arranged in a tetrad form by one membrane खाली 4 pollen grains which are present there. So this is about the structure of this. Now this is a vein or it is also the connective also. So here these 4 pollen sacs which are present, it is known as a tetrasporangium, and the formation of the microspore form the microspore mother cell, it is known as a microsporogenesis. So now you have studied here today the TS of the anther, or this. With the help of this you have to study the structure of anther. Now in this one thing that's very important, this type of the anther, it is a dithecous anther. Then monothecous anther which is also there in some species of the angiosperm. Only the 2 pollen sacs which are present there, that is known as a monothecous. Now here the 4 pollen sacs which are present here, it is known as a dithecous anther.